Good morning, everyone. I would like to thank the organizers of the meeting, particularly Melissa, for setting up not only the meeting but the consortium. It's been really wonderful for people at the country level to have access to the resources and people that have been amassed from this consortium. So just a bit of background on Lassa. It's a viral hemorrhagic fever that's endemic in West Africa. And it's got some pretty high mortality when you look at hospitalized cases. In Kenema Government Hospital, where our research is based, we're seeing 69% case fatality rates. The reservoir is Mastomys natalensis, a rodent that's endemic all throughout sub-Saharan Africa. So one of the big questions that we have, myself as a public health researcher, is where are people getting it and why? And some of the findings from the consortium that others will be talking about are helping us to answer some of those questions. So mastomys occurs all over sub-Saharan Africa, and yet we're only seeing Lassa in West Africa. And it's looking like it could be a subspecies that's permissive to the virus. And it looks like most of the cases are coming directly from the primary source, rodents to human, which have huge implications in terms of preventing the disease. So the consortium is actually a really wonderful venue for looking at some of the questions about Lassa fever from a more refined as well as holistic perspective. So we were very interested in looking at issues of land use and the impact of land use on abundance of small mammal communities, as well as specifically Mastomys natalensis, but also looking at livelihood practices, ecosystem services that increase or decrease exposure to Mastomys natalensis, virus regulation, but also the local knowledge that can influence or impact these dynamics. So this is our location. It's in Sierra Leone. We selected 4 villages in a really highly endemic area in the eastern part of Sierra Leone. These are our 4 villages. They varied in range in size. And for the larger towns, we actually only took a section of it in a peri-urban area. Mastomys is very common in villages, but it's always associated with agricultural communities. And so we really wanted to try to tease apart this connection or what's going on between the actual fields and agricultural activities and what's happening in the communities themselves. But the agricultural system in West Africa, in Sierra Leone specifically, has very spatial and temporal dimensions. So for example, this is one of our villages, La Leone. Has a very, very well-defined village where you have abundance of mastomys tends to be higher. Then you have tree crops that surround the village, and these are secondary forest. It's intentional planting of palm, cocoa, as well as shade trees. And then also you have, in low-lying areas, you also have a monocrop of rice that's farmed. And then in the dry season, it's converted into vegetable gardens. And then in the upper regions where you have good drainage, you have a farm and fallow system where you have mixed crops that are planted for about 1 to 2 to 3 years, and then fields are left fallow. So in terms of the temporal dimensions, they're very prescribed spatial dimensions, but also time points where there's specific activities that are tied into both the swamp farming and the upland farming. So we wanted to capture this in a 2-year longitudinal study with data collection at 8 different time points. Unfortunately, Ebola happened, and so our teams were quickly diverted to Ebola response instead. But we did capture, not longitudinally, but 4 key time points and distinct time points that I think are informative, and so we'll present some preliminary data at least. So we're using trap success as a relative measure of mastomys abundance here. But if you look at where mastomys are and when, you can see that we have a peak here, the highest peak, and this is occurring in fallow land. So this is land that was left fallow and has now been cleared to be prepared for crop in the uplands. The next peak we see is in the swamps, and it is in the dry season, so this is when vegetable farming is going on. Then the third little bump we see here is again in the upland areas when they're doing a lot of soil preparation and planting. So if you want to tie this into some of the social science work we did, trying to figure out who's doing what and where and when, in that first peak, the fallow land, we're seeing some pretty strict gender divisions, with men doing a lot of the brushing, what they call brushing and clearing of the land and burning, and the women and children supporting them with food and water, but also collecting wood. In the vegetable gardens, it's very, very conscribed to females, and women use this as a means of independent and discretionary income, so it's very critical, and you only see women doing this. Let's see. Now, in our third little bump of mastomys abundance, with soil preparation, this is actually a mixed bag of men, women, and children doing the planting of the intercrops here. So in all of these, you're dealing with a lot of contact with the soil. Now, if we want to overlay this with the epidemiological data, this is from a previous study done at Kenema Government Hospital. It actually correlates quite nicely, where we see a peak of mastomys and adellensis abundance timed with this activity of clearing lands and vegetable farming in swamps. And then again, you see a peak of mastomys abundance at a time where you see a secondary peak, but smaller peak later on in the season. So this is one of the only studies that came out that has shown an actual gender difference or any kind of demographic difference between groups that in Lassa fever incidence. So I think that that's exciting in some ways, but I think anytime we're dealing with any diseases that affect zoonotic diseases, neglected diseases, or diseases of the poor, we really have to take it with a giant grain of salt because we're really biased in what we're capturing. So one of the things that we really wanted to do, but haven't done yet, is to try to bring in some actual human seroconversion data. So in each of those time periods, we collected some blood samples as well to see if someone during that time is converting from negative antibodies to positive antibodies for Lassa virus. And we're going to take a look at if these divisions can be tied into gender or age distributions or any specific activities. We still have yet to integrate the local understanding of rodent diseases and control. And Jesse Bonwit, who's here in the audience, actually just published a really amazing paper highlighting a lot of these understandings. The other thing that we are really interested in doing is looking at the virus itself in incidence in small mammal communities, not just in mastomys and adellensis, trying to get an idea of how species richness has an impact on disease regulation, and also issues of interspecific competition. So that is what we're looking at now. So in terms of key findings thus far, we're looking at peak abundance of mastomys that's coinciding with a lot of human-driven land perturbations. And this is very gender and age specific. This also coincides with Lassa fever incidence, which was really nice. And I don't think always happens when we do this kind of work that it correlates so nicely. I think this is really an important study in terms of the importance of Lassa fever, which has been commonly associated with the biosecurity focus. But really, I think it's best understood in the context of an ecosystem service and the context of a One Health system. Mastomys are used not only, they're not just pests, but an agricultural pests, but they're actually used as a protein source. So there's a lot of cross-cutting that can occur, and so a lot of opportunities for some more research, as well as research that might have an impact. Thank you.